Yeah, I think we are ready to start, or Christoph is already ready to start. So he will talk about structuring, analyzing, and harvesting big data and material science, electron microscopy. And welcome, Christoph. The floor is yours.
Yeah, thank you very much, Claudia. Good. Yeah, so I'm actually very happy for this introduction that Claudia just gave, and also for the talks of this morning, because they really allow me to not have to explain everything that I would otherwise have to explain, but jump right into what I wanted to talk about. And as Claudia has said, I do not, or we do not, or my group does not try to create its own database, but we like to contribute to this project that was mentioned, and that is this FAIR-MAT project here that Claudia is the spokesperson of. But I will say a little bit more on this in a minute. But the focus will be, as the title already says, on material science electron microscopy. I focus on material science electron microscopy because there's also life science microscopy, which is a different universe. And there's also different databases and so on that I won't talk about now. Okay, so yeah, what is the motivation and what are the obstacles for fair data infrastructure in experimental material science? Of course, a number of motivations have already been mentioned, and they have been established already when this acronym FAIR was established. We all want to share because we benefit from it, because we can reuse also other people's data, of course. And one of the applications for that is that we can train artificial neural networks to somehow annotate or interpret data for us. And this interpretation is often then based, of course, also on already existing data. And for that, we need these databases that, of course, have well annotated data in them. And you can obtain this. Claudia has mentioned this example already. For example, by high throughput combinatorial experiments, and we've heard a nice talk in the morning by Christoph Babitz on this. You can have databases of experimental data collected by many different labs. This is what FAIR-MAT wants to establish, and I will talk more about this. This is actually quite complicated, and I will show you why. But you can also, of course, if you have already a theoretical database, you can simulate experimental data if you have a very good forward model, like a digital twin of your experimental setup. And that is actually also a very valid way of doing this. If you have a good model, if you want to establish this connection between theoretical data, on ab initio, for example, and other, for example, atom positions, and your experimental observations, then actually doing realistic simulations is really one thing to invest into. Because then you can really compare theory with experiment on a one-to-one basis. Of course, having databases and fair data allows us to also crowdsource data generation, especially in this second item here, where we then use data from different labs. We can also, of course, crowdsource then the knowledge extraction of data, because some people may not even want to collect data themselves, not do any experiments or any simulations, but only interpret data that is available. And it immediately then becomes available to the rest if it's done properly. But also data analysis workflows. So just how to process data, how to make sense of it, how to interpret it, can then also be put basically in this common, on this common basis, and everybody has access to the most recent techniques. This is what I want to say here, that data analysis workflows can immediately be applied to huge amounts of data if they're available, and people can then, of course, benefit from these data analysis workflows that are available. And with that, you get accelerated growth of knowledge in material science, of course, because we don't have to reinvent as many wheels anymore as we do at the moment. And there's also innovative approaches of AI going back to collecting more data, because now you can, for example, have algorithm and data-driven data acquisition. Right? We've heard that in the talk of Christoph Babitz, where there was an optimization algorithm that was trying to find an optimum feature in the material, and that was driven by data that was acquired within this high throughput experiment. But you can also think of using data that is on a common database that can drive these experiments. But as I said, there are also obstacles that we have to overcome. First of all, we have to convince the community. Not many of our colleagues are convinced. We don't have 10,000 people attending this conference right now because not as many people want to invest into stuff that is not related to the actual scientific question that they are actually working on. Then we have a lot of incompatible data formats, proprietary data formats, where we don't know exactly what's in the files, what companies that develop instrumentation store in their data files, or how they store the information. Then we have also incomplete or imprecise metadata. So often our instruments may not be as well characterized as, for example, the parameterization of an ab initio code or so, or some MD code that calculates some atomic structure or electronic structure. And also, even if we have characterized this very well, in many cases, these instruments are not digitally accessible. So we cannot read out the parameters that we would need. We would have to copy them by hand and so on. So all these things are obstacles. And also, especially when you have a complex experiment, you often invest a lot, and we do this ourselves as well. We invest a lot into somehow making some, drawing some physical picture from the measurements that you have, and you do some very complicated data processing. And often these data processing steps may not be irreversible, so you may not actually be able to undo and go back to the original data. So it's always important, of course, to have the original data. And it may not always be easy to really reconstruct that link that some research group has established between the experimental data they collected and the publication that they got from it, and so on. So there are many other issues that you can think of that may be obstacles in terms of having a fair data infrastructure in experimental material science. I just want to say quickly, our approach that we work with, and just so that you understand, this is the background that we come from. So we operate microscopes, so often, mostly electron microscopes, but also optical microscopes and other instrumentation. We invest a lot into this topic of data reconstruction. So reconstructing, for example, 3D atomic positions from experimental TEM data, but also modeling our experiments, once we have a model of, for example, an atomic structure or an electronic structure, that we then model the behavior that we would see in experimental observation, so that we can really compare on a ground truth, basically on an experimental data level, what we would expect from our model with what we see in the experiment. And that actually is, I would argue, something that is really very important to pursue, that you have a forward model. Often the reverse is not so easy and may then be potentially reconstructed once you have a forward model, but the forward model is often easier, and it allows you then really to compare data on a basis where the statistics is correct. So often, especially in our case, where we have limited number of counts, limited number of electrons that we detect, there are certain Poisson statistics, and if we do some processing through the experimental data, these Poisson statistics are somehow warped and they cannot be interpreted in that manner anymore. So any statistical comparison of data would have to actually really be aware of all this, which is often not really easily possible. So to compare apples with apples, we would really have to go back to the experimental detector level from our model that we have about what we expect to see. And then we can adjust the model to make it fit the experimental data once we have a good digital twin or forward model of our experimental setup. Here's just one example of how data is often treated, experimental data. Just an example from a former PhD student in my group who is now in Switzerland at EMPA, working with Wolf Erni. And he developed this modification of a UNet. UNet, probably many of you have heard, it's a tool to process images, and so, for example, you can use it for denoising and so on. And he made some modification that converges faster, but he also applied it to various types of data. So this is not electron microscopy data, as you can see, but he did a simple experiment that didn't require him to have access to an electron microscope. So you can see here, this is a laptop. This is the screen of a laptop, and the laptop actually faces the door of his room. And you can see the reflection of what plays on the screen on this in the door. And we as humans, we cannot interpret very much from this. I mean, we see that there's some speckle, but not really much content. But if you train this MNet that he developed with this data that is somehow reflected from the door with the ground truth that is played by the screen, he can then actually record only the signal on the door and recover most of what you would see on the screen. So a movie plays and you can somehow capture the story. So some information is lost, but it's amazing how some of these computer algorithms, if they're trained with plenty of data, can actually recover a lot of information from very limited amount of data that is actually available, or very noisy data. So here is another example. Now, this is not a laptop image anymore. This is now recorded on an actual transmission electron microscope. So this is some atomic structure. And the question is now, where are the atoms and how many atoms are there? What's the size and the structure of this atomic cluster? And applying basically these denoising techniques, you can now really retrieve where the atoms are if you have a good forward model of how this works. So you need to take structures like this and then apply the correct Poisson statistics and instrument parameters to simulate images like this. And that is how you establish this forward link. And then these neural networks, they can actually learn how that works and can do the reverse step for you, because that is often not really easily possible, at least not in a deterministic manner. But neural networks are very good in doing this. So you can also use this type of technique to, for example, if you have defocused images of cells. So if you've ever looked at cells in an optical microscope and you saw not much contrast, but when you then go out of focus, you see some contrast. Well, that is due to the fact that these cells may not be stained, and actually there's only a phase shift. But neural networks, if you give them images of these types and give them also the ground truth, and you can obtain the ground truth by some advanced techniques like holographic techniques and or focal series reconstruction or so on, then you can train these networks to actually interpret these images. So using only 2 images, you can actually get a very good representation of this, and using more images, you can get an even better representation of this. So having all this basically available online in a common database for experimental data, all these tools, you don't have to reinvent them again in every lab, but people across the globe have access to these. And it also benefits the data infrastructure itself, because once people realize that this is a useful thing to have, they will also voluntarily upload their data to be processed by these tools. And with that, feeding the data infrastructure itself to have even more data to base its results that it produces in the future on. So this is somehow, I guess, the way that also Google works, right? You provide some nice software or some nice tools that people find useful, and they voluntarily provide everything about their life to the infrastructure. Here, it's not about your life, it's just about your data. So here is just to complete that story about the atoms. Of course, then there's more advanced tools that other people have developed. And if you make all these then available to the database, that's even better, right? So this is a tool developed by the group of Sergei Kalinin at Oak Ridge National Lab. And they, instead of only extracting atom positions, they also extract the reliability of these atom positions. So you can, if you have enough data, and you can then run different reconstructions with different seeds and different parameters, you can then also get some information about the reliability of a reconstruction, something that, for example, we wouldn't have. But once these algorithms are shared on a common database with a common data format that processes our data very easily, then we can immediately also gain information of this type here. So there's a lot of benefit in sharing these algorithms and also the data. Here, one last example I wanted to give is Tychography. It's a compute-intensive technique where you have your sample and you position your beam to different positions across your sample, and you collect diffraction patterns at each beam position, and then you can reconstruct using some basically a layered structure that's like an artificial neural network that at the bottom basically produces your experimental data, but within the structure, you have now the three-dimensional potential that scatters your electrons. So there's a very nice physical representation of this actually. And then you can reconstruct the layers of your material that have scattered your electrons. And once you implemented that, you can even now train the computer to control your microscope, to position the beam to where you would most efficiently expect more data to build the structure. So this is just an example to show you that having these data-aware algorithms or data-trained algorithms, you can now even feed back into the experiment and drive the experiment based on basically the data that you have already collected prior. So this is one of these tychography reconstructions obtained in our microscope here in Berlin. And this is a very simple material. It's a monolayer of molybdenum disulfide. And so many diffraction patterns have been collected, and then the reconstruction algorithm has run and produced this beautiful reconstruction of the atomic positions. Now, there is, of course, a trade-off. You don't want to position the beam too closely and collect too many diffraction patterns for too long, because you may damage your material. And there are these techniques called compressed sensing, where you then only position your beam randomly and try to then still do reconstruction with that information. And this would be a reconstruction from a compressed sensing acquisition scheme, where you have random positions of your probe, and you only have a factor of 40 less probe positions. So your material doesn't see as many electrons, it may survive longer, but your reconstruction is actually not very good. But if you now let the computer learn while you're doing the acquisition to position your probes more smartly, and this is a pre-trained network that has been trained with experimental data, it starts first randomly, but then it improves and gets better and better data. And of course, this has to be pre-trained. I mean, this acquisition here, that was relatively fast, but the information about how to interpret these diffraction patterns and so on, that had to actually be pre-trained and took 2 weeks. Now, if we could make this available online in a shared data infrastructure, immediately a number of people could benefit from this training, and it wouldn't have to be redone. So just a few motivations for why there's great potential in having a shared infrastructure for experimental material science data, but also data analysis workflows and knowledge extraction from data. And I promised you I would get more into this topic here. So we can do this classical ANN training, and I've showed you a few examples of how these artificial neural networks can help. Now it's not so difficult to do this in high throughput experiments where you are in control of all your parameters. Or if you do realistic simulations, then again, you have control of all these parameters. But if you acquire data from different labs, well, you don't have as much control, and a lot needs to be done actually to make this happen. But there is again great potential, because now you can apply this crowdsourcing idea here, that you don't have to do everything yourself, but you can benefit from what other people do. So here's one example. Again, artificial neural network processing data. This is an example actually given to me by Stefano Cosini from Trento, but within this NFFA Europe project, where they have an infrastructure that provides access to microscopes and other setups for people who don't have these instruments themselves. And he actually, from these many investigations, he had 22,000 images and was able to have some students train this machine by annotating some portion of these 20,000 images, and then the test data set then was able to show that images can automatically be labeled. For example, you have particles, or you have tips, or whatever. You have a biological sample, and so on. The thing was that all this data came from the same instrument. So this was one and the same SEM that collected the data always with exactly these parameters, and these could all be retrieved. So the magnification was calibrated and the current and so on. So these images could easily be interpreted. Also, the detector was always the same configuration. That's very important in electron microscopes, for example. How do you detect your signals? Here's another example. This is when you shine in TEM, electron beam through a sample, you can collect the fraction patterns. And the fraction patterns, if you collect them different ways, they may look, for example, like this. If your convergence angle is smaller than the inverse of the unit cell size, these disks don't overlap, and you get these beautiful patterns. And they contain a lot of information. But one thing that people want to extract from this is how thick is the sample. So you do a lot of simulations, and then you train an artificial neural network, and you can then, these are these position average convergent beam electron diffraction pattern. So you position average over basically one unit cell in your material, and you get these patterns, and you can easily compare that with simulations. And it has been shown that these are very sensitive to the thickness, for example, these patterns, and you can then extract very precisely the specimen thickness, which you need to know for further analysis of your data. So again, this neural network needs data. It needs to have input from many different crystal structures and could be then shared online with others. But if you do this for the same experiment, for the same microscope, you can easily do this because you have calibrated your camera lengths of the diffraction patterns and so on, and also the way that your detector works. But if you try to apply this now to data provided by other groups, it becomes more complicated because the detector characteristics play a role. Maybe the calibration of your camera length needs to be redone and so on. There are many complications, and I will deal with that right now. And you don't even have to go across the country or the globe to have data from a different lab, even within the same lab. So this is our physics building here. This is the Iris Adlershof. And even within this building, there's a number of different microscopes, actually from 3 different groups that somehow pool their microscopes here together. And comparing data from any of these 2 microscopes is already a challenge. And so we already have a nice playground locally here that we try to tackle. And once we make some progress with this, we can hopefully then also very easily go beyond the boundaries of our city or basically this local lab here. So if you look online and look for databases on experimental data related to what we can look at with electron microscopes, you can see, for example, and I think it has already been mentioned in a talk today, for example, this Crystallography open database, where data is stored in a format that can easily be read, and that is actually independent of instrumentation. So while in this database, this is, for example, the EELS database, and we'll come back to EELS, and Claudia has already mentioned EELS as an example in her introduction. There's raw experimental data. In this database here, now we have crystal structures. That means atom positions. And this is like theoretical data that you can now apply a forward model to compare your experiment with. Actually, this is instrument independent. So it's actually much more easy to use and much more widely applicable than this one here, which provides raw data that is instrument dependent and may not even be interpretable at all, because some of the metadata that would be required to do the interpretation is missing in this data. But I will come to this example of EELS here in a minute. So this is some data that Alberto Efferat from my group, he acquired on our microscope, which has a very good resolution in EELS. So this is the zero loss peak for anyone who knows about EELS. It's monochromated, so it has a very well-defined primary beam energy. And this is the plasmon peak. But what we're interested in is, can we map the band gap in silicon? And that is always a challenge for these electron energy loss spectroscopy experiments, because the band gap of 1.1 eV and the very weak signal makes it difficult to somehow separate from this zero loss peak. So this what you see here, this is the zero loss peak. That is in principle this very sharp peak that you see here, only blown up. Basically, this small area down here is blown up. So he then developed this reconstruction algorithm. This is also something that is computationally quite demanding and runs on GPUs and takes a while to process multiple spectra. But it basically tries to now self-consistently reconstruct the dielectric function of this material, taking into account the thickness of the material, the primary beam energy, relativistic effects that somehow produce artifacts in these spectra, to then retrieve the dielectric function. So this is the dielectric function represented in N and K. These dashed lines here are from the database. I have a link to that in the next slide. I forget what name. It's dielectric function database. So this is from optical measurements, the reference data. It doesn't quite look like the reconstruction that we obtained from these experiments, despite this advanced reconstruction algorithm here. But the assumption was that you have pure silicon. And of course, now you see already the problem that we have with experimental data. We always make assumptions about the data. And that is the assumption that we then also write as metadata, excuse me, into the database, right? This is again the picture I've shown you in the previous slide. This is now not in the N and K representation, but now in the real and imaginary part of the dielectric constant or dielectric function as a function of frequency. And you see there is not a very good correspondence to the optical reference data. But when Alberto now assumes that there is also native oxide on the silicon, which makes sense if you know how the silicon sample was stored, right? It was not processed basically under protective atmosphere, but it was exposed to air, and then the native oxide can grow. And if you have that information, then you can also make that assumption, and you can then build that into the model. And now actually reconstructing the dielectric function from this material looks much better. So now you have here the solid line is the optical reference data, and the dashed line is the yields reconstructed dielectric function, and you can see much better agreement. So what I'm saying is many of these experimental databases, for example, this yields database that I showed you before, may contain data that is not useful at all, because you don't know how the data or the sample were processed. And that's why it makes it very difficult to actually work with if you don't have all the information. And one really needs to have, sorry for this. And one really needs to have very good information about how data was obtained. And now I come to the project that Claudia has already mentioned, this FAIR-DI project, and she has not said very much about it, so I will say a little bit more about it, and also Balk Ansdorfer has mentioned it. So it's a large project. It includes many partners spread across the country, Germany, because it's a response to a national call for data infrastructure. Claudia is the spokesperson, Matthias Scheffler is the deputy, and there are actually many institutions that participate, also large-scale projects which produce lots of data. And it's split into these 7 areas where some of them are administration and also use cases and so on, but 3 of them are actually more focused on really how data is being collected, or areas where data is collected. So one of them is synthesis, so different types of synthesizing materials. Another one is experimental materials characterization. That's what I'm going to talk about more. And then the third one is also computational material science that is represented by Claudia especially, but there will be a talk also by Markus Scheitgen on this, as far as I know, and it's based on the whole project actually lives and actually thrives due to the large amount of work that was already put into this NOMAD database that already exists, and that contains lots of theoretical, especially ab initio data on atomic and electronic structure of materials. The experimental science part, that is now the characterization of materials using different techniques, does not want to cover all different aspects of experimental material science characterization, but wants to focus first on only 5 techniques. So these techniques include atom probe tomography, electron microscopy, angle resolved full electron spectroscopy. You've heard about a talk by Ralf Ansdorf already, optical spectroscopy, and also XPS, so photoelectron spectroscopy. And they are very diverse in the type of data, the data formats, also the instruments that they use. And so if we can tackle these 5, we hope to be able to actually extract this also to other data acquisition techniques. So what we now focus a lot on is how do we actually allow data to be collected and made available to databases like this. And we need to start actually where the data is being generated, and that we facilitate the digital acquisition of data and also incorporation into databases. And we work very heavily with electronic lab notebooks on this, because they actually allow us to then have data sets that we can transfer to a database. And so in electron microscopy, we need to prepare samples in a specimen preparation lab, and these samples are then already recorded and characterized and so on, and information is then processed using such open source software that can do image processing, that we can run the same software on different machines and operate optical microscopes and also electron microscopes with it. And data is then pushed from there into the electronic lab notebook, where it can then be further processed and annotated and so on. And once we go to our microscope, this is actually a TEM. This is how the software is used operating the SEM. The same software can operate both microscopes. We draw this data from the electronic lab notebook into here, so that we have information available about where the sample came from and so on. And we can then process the data and push it again to the electronic lab notebook. And the same happens when we then process the data in our office. Actually, I will show you that the data processing can even be done directly on the electronic lab notebook through a web browser, which allows actually much faster sharing of data analysis workflows and so on. And you can also make it more reproducible, because you have better control over what happens to your data if the processing is done in a controlled environment. So actually the same software here that you can see here that controls the microscopes and we can analyze the data with, can also now run actually on the server, and we can operate it through a web browser, which really makes things also much more easy. Also, when training new students, for example, they can train basically online and don't, all the tools are available to them right away. And we use an electronic lab notebook, and we wanted to have something that we can easily modify ourselves, and that can also be extended by us, and where we can also give it away to others and recommend it to others without asking them to pay a lot of money. And so we decided for an open source solution that was developed by Nicholas Karpy at C-Lab FTW. We discovered that actually independent of us, also other labs have adopted this solution. So we developed solutions for this, but keep in mind that we can also copy them also to other eLab or electronic lab notebook solutions. But this is basically the platform that we currently build on. And also very large data sets that you would not normally be able to download and process on your own laptop can be then operated and processed now and viewed and browsed and so on basically the server. And that is another incentive for us to actually work with this, because some of the data sets that we collect, they're huge. They're very large. For example, here's a data set that has 34 gigabytes. That exceeds the RAM of some of the computers that we have, but you can now process these data with your local laptop very easily. Right, and for this to happen, you have to actually bring now these different solutions together. I've heard already one of the talks this morning, hackathons being mentioned. I think this is something that in this process of developing these data infrastructures is something very important. So we got together people from the LiberTem project. So this is from the Research Center Jülich, who developed a software to browse arbitrarily and browse and process actually arbitrarily large data sets. Then we get also somebody from the Neon company who provided this open source graphical user interface for processing 2- and 3-dimensional data sets. And then we have our own specialists. In particular, I want to highlight here Shajir Shabi, who really invested a lot into this and was responsible for much of the software that I'm going to tell you about. And of course, also you have to have the users, those people who want to work with this and have applications of this and help to actually develop publications from these developments so that you can have joint papers at the end that also have scientific relevance. So I see that my time is running out slowly, but I won't get into all the details because some of these may be a bit too specific to microscopy, but I have 3 little videos that I just wanted to show you. But before, let me just show you how the data infrastructure that we're working with works. So we have these different instruments that collect data and that are able to now push this data to the electronic lab notebook. Either you do this through the web interface of the electronic lab notebook, or you do it directly through this data acquisition software that I've shown you. One of the videos I may probably skip, but shows how you can directly from the data acquisition tool. I think this is essential actually, that many of the data acquisition tools that people work with in the different labs, that they somehow extended using plugins or whatever is possible with this software to directly push data to, for example, an electronic lab notebook or a database and so on, because you want to lower the threshold as much as possible to ingest data into these common databases. Be it only your local database from which one can then copy data into a common database or electronic lab notebook or whatever, but you want to have it somehow in a place where it can be sorted and annotated and so on. Because if that step is not done and if that step is not easy, it will just end up on some hard drive in some PhD student's drawer. And after the PhD student has graduated, nobody will ever know what to do with that data again. So this first step of getting your data directly during the characterization session into the database, electronic lab notebook, whatever, in our case, it's this electronic lab notebook, that is actually very crucial. And that is actually where we saw we have to invest first, because once that step is not done, we don't have any data to actually process and do other things on, right? And of course, there needs to be some permanent storage because you want to keep the raw data that needs to then also record it in an immutable manner so that people cannot modify it. Users can interact with it. And what's now very important for us, where we really see a big benefit and where we also see that people may like this idea and may want to actually process their data directly on the server, is that we provide tools that make it easy and also provide methods that they may not actually have locally on their computer available. For example, we have also a server that has 7 V100 GPUs. So this is not something that you have on your desktop. So you have to apply some of these methods on the server anyway, if you want to use them. And this eLab FTW server actually gives access to all this. So it runs Jupyter notebooks on your data. So you're very flexible in the way how you process data. You can also have this tool, Liberten, that can browse and process huge amounts of data. This is actually linked now to also this open source GUI, where you can have any user interaction that you want with your data when you process it. And of course, also inline tools. For example, we have focal series reconstruction and some atom probe tomography tools and so on. And then also other tools that you normally have and that people have developed tools with, that provide now access to this data directly on the server through a web browser without having to install anything on your local computer. So I see my time is already welcome. Sorry, coming to an end, but I want to demonstrate. If you have any questions on this, I can definitely answer them, but I want to show you one last video, and that is now how to actually run different applications in this electronic lab notebook. So this is just a demo setup where there's only one entry in this electronic lab notebook, just to demonstrate this capability. So you can now edit this and you can go into it. There's not much data yet in the description of the experiment, But you can have, for example, now Jupyter notebooks uploaded here. And if you click on this Jupyter notebook, you get access to all the files that are within this experiment that you collected. And you can now run these Jupyter notebooks and process data. And you can also have now, again, another tool that's this Libertem that can browse these very large data sets. And again, it opens a new window, and you can now have this other tool that can now work with your data. And you can now see the different data sets that are available within this experiment. So one thing is, of course, you need to have a container that makes your code only accessible to the data that corresponds to this experiment. So here is, you can also do a reconstruction. So this is now running a reconstruction. This is, again, a Jupyter notebook that opens one of these data sets. And it's a HDF5 stack that you can now really modify online within the browser, this Jupyter notebook. And, for example, display different frames of this data stack. It's a focus series. So nothing super fancy, just to show that it can be done. But what you can also do now is you can, if the file has a certain extension, you can now actually even, the software knows that this can be associated, for example, with this reconstruction algorithm. Data is structured in JSON files. So if you want to have any structured metadata, you can have free text descriptions, but also structured metadata in these JSON files that you can browse and edit. And if you now click on the reconstruction, you can see it here. Then this reconstruction runs directly on the server. And once the reconstruction is done, it actually then ingests the reconstruction result that was obtained back into the electronic lab notebook, so that you now have also the reconstruction results available on your server as part of your data set that you've created. And I guess this will be now you refresh the view, and you can see there are now 2 new data sets that have been added to this entry in an electronic lab notebook, which are your reconstruction results. So I think I won't show you everything, but I want to finish now with a summary. So there are a number of obstacles for implementing FAIR principles in experimental material science, especially when you want to combine information from different labs, because you have data formats, you need to record metadata in a digital manner, and so on. But once you have that, machine learning can really give you a huge amount of benefits, and these benefits become immediately available to everybody who participates in this project. And I will stop here, and thank you very much for your attention.
Thank you, Christoph. We still have a few minutes for questions, so you could either speak up directly under your microphone or write a question to the chat. I don't see any hands up. Also nothing in the chat. Let me ask a question meanwhile. So you mentioned already the different solutions, different file formats, all these kinds of things. So of how many bigger, let's say, initiatives are you aware? Are you aware of bigger initiatives who are doing similar things? Because obviously now the time is ripe, everyone tries to harvest data, to verify data. So obviously there are many things done now in parallel. Are you aware of who is doing what?
A little bit, but I won't be able to now give you a complete list of what is going on. I would have to sit down and really think very hard, and also maybe do some research. But yeah, definitely, there are other activities. I mean, even in Europe, we have the European Open Science Cloud, where a lot of projects, including FAIR-MAT, also will ultimately, hopefully, link into. But for example, today I learned that within Esteem3, which is one of the activities, it's a European Network of Excellence where people have access to TEM infrastructure. They have also something where they provide an online processing of diffraction patterns and simulation comparison to, for example, do this PAC-BET analysis that I showed on this position average convergent beam electron diffraction analysis. And yeah, so in the life science, for example, there is already a large database in this. They are much better organized because I believe they're also much not as diverse. Life science electron microscopy. They have a well-established database already of electron microscopy images of cells and proteins and so on.
Do you have an idea how many electron microscopy groups are in Germany or Europe or so? Because it's a very popular technique, also being a very expensive instrument. I would be curious to how many people one would have to pick up if you want to homogenize the community.
Yeah, even in Germany alone, you would have probably at least 25 TEM groups. I mean, SEMs you can find in pretty much every lab because they're easier to use and cheaper. But even TEMs you have, well, again, I would have to think about it, but roughly 25 at least, yeah. And then of course, in all the other European countries, and you have a lot more. And then across the world, in the US, you have a huge number.
So Matthias has raised his hand.
Yeah, actually, a question which, in fact, I had asked you, actually, you had explained to me several years ago, and that was mentioned by you, but also by others, the problem of proprietary software of the companies. Now, I think you have some experience how to convince companies to get more open, which I think is really very important. Can you elaborate on that?
Yeah, so I mean, we do talk to companies that open their, and just really different companies have different company spirits or philosophies or whatever, so they are more or less open to this. But I know, for example, and this is, I think, a good example from the life science electron microscopy community, they have decided on an open data standard. And they have basically agreed that all data should, so everybody buying a detector would have to convince the company providing the detector that they actually save data in this format. And now even a company that has always had closed formats has one open format, and that is the one that these life science people have told them to include. And I believe now with all these open data formats and standards and requirements by funding agencies being around, companies realize more and more that they have to do something, and things are changing slowly. So I think we're riding a wave where it becomes easier to actually convince them.
Great, good, thanks.
Okay, last chance to ask a question. Okay, I don't see any, so then we are perfect in time. I thank Christoph again. And we are moving to the next presentation that is given by Volker Blum.